Welcome everybody to my presentation on the identification of a novel non-NLR leaf rust resistance gene. And the work that I want to describe started many years ago when we noticed that the Swiss winter wheat cultivar called Forno, which is shown here, has a durable resistance, and we started to make crosses and to do genetic analysis by crossing it with susceptible leaf rust winter wheat cultivar Arena. And already many years ago, we identified 8 chromosomal regions in cultivar Forno, which contribute to this durable resistance. One of the most important ones is of course the genome 7DS, which we later cloned and identified as LR34. Then there is a genome 1BS, which we identified as LR75. This is only genetically mapped, but not yet cloned. There are a number of minor QTLs. And finally, on chromosome 7BL, there is another major QTL for this durable resistance. And what I want to describe here is the identification of this gene. So here you have the seedling phenotypes of Arena and Forno. Both are susceptible. When you now introgress this 7BL region alone or together with LR75, you get these near isogenic Arena lines with LR14A or with LR4. I already called the gene on 7BL now LR14A because when we introduced it into the background of Arena, we discovered that the gene gives this typical mesothetic phenotype with different infection types on the same leaf, very, very similar to the phenotype of LR14A in the near isogenic line Thatcher LR14A. We also think that there is a suppressor in the Forno genetic background for the seedling resistance gene, and that's why only when we introduce it into this Arena background, we see the activity. LR14A has been genetically mapped earlier. It's also on chromosome 7BL, and that's why we thought it's quite likely that based on the genetic location and the phenotype that the gene in Forno is LR14A. Now, to identify the gene, we took this Arena LR4 genotype, we mutagenized it, and then we screened for seedling susceptibility. We identified 7 mutants, which are shown here. And then we took advantage of the fact that the Arena LR4 genotype is part of the 10+ wheat genome project, which is led by Curtis Posniak. And all these genotypes listed here, including Arena LR4, have been sequenced to a very high quality, which is equal or better than the reference genome sequence of Chinese Spring. All these genome sequences, including ours, is already available, but the publication of the work has only now been accepted by Nature, and it should come out in the next few weeks. Now, we used a modified MutChromSeq approach to get to the gene. We had this full-length pseudomolecule from chromosome 7BL from the Arena LR4 genome sequence. We had to synthetically fragment it to make it compatible for the analysis with short Illumina reads obtained for the mutants. Then we identified contigs which contained mutations in all the mutants, and we found actually one contig which contained a gene, a coding sequence, and which contained mutations in this coding sequence in all the mutants. So this gene that we identified on this contig encodes for 3 exons. Here are the locations of the mutations. They all change an amino acid or introduce a stop codon. And when we now look at the encoded sequence, then there is here this N-terminal domain, and followed by about a dozen ankyrin repeats, which are indicated here in blue, and then 5 transmembrane domains indicating that it's a membrane protein. To provide further evidence that we have identified the correct gene giving this seedling resistance, we used virus-induced gene silencing. Here you see a control of arena with a silenced phytoindesaturase gene, resulting in these chlorotic stripes. Here you have the wild-type RE9LR4 with a virus not containing a target sequence, and you see that resistance is not affected. However, when we specifically introduce a fragment from the cloned gene, we get susceptibility because of the silencing. So this provided further evidence that the cloned gene is actually the gene that gives resistance in RE9LR4 at the seedling stage. Further evidence also came from genetic mapping, where our cloned gene co-segregates with markers which have been described in mapping populations to be very close or co-segregating with LR14A in earlier publications. And the gene was also present in the donor lines of LR14A, such as Hope's and Solkirk, and Hope was actually the first bred-wheat genotype where the gene was introduced into from Emmer wheat, where the gene is originally derived. So with these three lines of evidence, we are quite confident that the gene is actually the one which confers this resistance. Now, the resistance of LR14A is race-specific. It's a classical race-specific resistance, but obviously it is encoded by a very different sequence. The gene is expressed only after infection with an aberrant leaf rust isolate. As you see here, non-infected seedlings do not express the gene, and there is this induction of after infection. This induction is not observed when we use a virulent isolate on LR14A. As you can see here, there is no significant induction here at 6 days post-infection. And so this clearly shows that there is a race specificity of induction of the gene. When we look at the genomic level, and compare the LR14A region in RE9LR4 together with the Chinese spring region, we see that there is good collinearity in the genomic region, with the exception of a piece of a bit more than 200 kb, which is simply deleted in Chinese spring. And this haplotype, this deletion haplotype in hexaploid wheat is actually quite frequent, and obviously in Emmer wheat, where the gene comes from, there is some additional sequence here. The protein is localized in the plasma membrane, as is shown here in these experiments, where after plasmolysis we have this very nice localization in Hessian strands and in nanodomains of the plasma membrane, confirming the location in plasma membranes. The schematic model of the protein is shown here. This would be the amino terminal domain, the ankyrin repeat domain, and here the plasma membrane location of the C-terminal domain. Here would be the Raptor X prediction of the ankyrin domain of LR14A. When we did an HH-spread analysis of the LR14A protein, we were quite surprised that the best homologs are these transient receptor potential cation channel TRPA1 proteins from Homo sapiens. And these are actually calcium permeable non-selective cation channels, which allow calcium to pass the membrane. And so our current model is that LR14A encodes a calcium channel, of course only when it's present after infection with an aberrant phenotype, and that this calcium channel is somehow regulated by the ankyrin repeat domain. Now, to summarize, LR14A is a race-specific leaf rust resistance gene, originally introgressed from cultivated durum wheat. It's race-specific. It confers this modifier-dependent mesocetic phenotype. We cloned the gene. It's specifically induced by an aberrant isolate, not a virulent one. It's a plasma membrane protein, and it encodes a novel type of gene involved in race-specific resistance in plants, and possibly this is a novel class of resistance genes which is useful for agricultural use. And I want to thank Marcus, Simon, and Thomas in particular, who did the majority of the work, and all the funders and collaborators which made this work possible. With this, I would like to close, and thank you very much for your attention.